Okay, so welcome back to the first day afternoon session. So today is more of not really, it's more of introduction about the laboratory, the skills or tutorials. Is it okay?
Yes.
What's your name? I forget. Your name?
Abhinek.
Abhinay. Okay. Uh, so as I said, myself Raju Gopal. I'm professor here in the Department of Plant Science, and our laboratory, maybe now we can go through later the profile of lab. So we work mostly on photosynthesis aspects and abiotic stress related the proteins, the role of proteins or the organization of thylakoid membranes under various stress conditions. And we were been working on state transitions where photosystem 2 and photosystem 1 there is a balance. The excitation energy has to balance from photosystem 2 to photosystem 1. So that balance is called state transitions. Through light arresting complex, they migrate using a kinase called STT7 kinase, STN and STT7 in Chlamydomonas, STN7 in Arabidopsis and higher plants. So recently we also started CRISPR technology to editing some of the genes to understand the photoprotection mechanism, which is on thioredoxin and also autophagy related genes, and connecting again through more of molecular biology and physiology, biochemistry to understand again photoprotection is the main our goal, and also want to understand the lipid role in lipid accumulation in various stress conditions using CRISPR technology. These are some of the areas. Anyway, I am going to talk from tomorrow onwards about my research. So, as I said, I have about over almost 25 years of experience in this. I mean, if you say that from the my PhD days, it's almost like 30 years of experience on photosynthesis related aspects. Started with, as I said, I also started cyanobacteria as a model organism. Then worked on various algal and plant systems. So this is about me. As said, HRO we have already introduced. I think it may be introduced much better tomorrow in front of the Vice Chancellor. Where he's, as I said, he have over 40 years of a research experience in photosynthesis, and mostly in plant biochemistry and physiology and molecular biology. The lab is mostly focused on molecular biology, where protein-protein interaction. Anyway, he is going to talk about it himself, so that it will be more interaction. So, one round of introduction yourself. So then, then we'll go with the tutorial thing what we are going to do. Okay. So now, yeah, maybe we can start from this side. You can sit and tell, or you can stand so that everyone can see. You can just switch on that one, middle. Yeah.
Good afternoon, everybody. Myself Subhashri. I am from Utpal University, Odisha.
So what are you doing? MSc or PhD?
Yes, I am pursuing MSc in botany.
Oh, okay.
Good afternoon, everybody. My name is Sharmila. I am from Odisha. Currently, I am pursuing MSc Botany in Utkal University.
Myself, Ramsurtidas, and currently I am pursuing MSA from Utpal University, Odisha.
My name is Satyajit Kujul. I am from Odisha, currently pursuing MSc Botany in Utpal University.
Myself, Dr. Madan Shekhar Kalwa, assistant professor at the University of Vizag. My major research area is mostly like photosynthesis and changing climate, research to different types of food crops, most likely higher
and will be also one of the resource person.
Good afternoon, everyone. Myself Pranti Pradhan from Odisha, currently pursuing MSc in Botany in Utkal University.
Which college? Which university? Utkal University. All of you are from Utkal only.
My name is Akshay Kumar Desai. I am from Odisha and currently pursuing MSc in Utkal University.
Maybe we can take.
My name is Kunjal. I am from ISA Tirupati. I am pursuing PhD and right now I am a visiting scholar at University of Hyderabad.
Yeah.
I am Nandana. I am just completing my Masters in Biotechnology from Pondicherry University.
Good afternoon, everyone. I am Vishal Jamne, PhD scholar from Goa University.
Good afternoon. My name is Tanuja and I am from Geetam Visakhapatnam, pursuing PhD under Dr. Madan Shekhar sir itself. So I am working on plant science and majorly pigeon peace response to abiotic stress and how it is tolerated. Hello, I am Lalit Balkis. I am from Kerala. I graduated my MSc last year, and for the previous 1 year I have been trying for the CSIR net coaching.
Myself Shrija. I have recently pursued my MTech from JNU and now I am attending this workshop to know more about fluorescence filaments.
Celia, you can start from.
Good afternoon. I am pursuing my PhD. I mean, currently in 4th year. Good afternoon, all. I am currently pursuing my 4th year of PhD in Professor Rajgopal sir lab. Currently, area of my work is under photosynthesis at drought condition in a plant and algae as a biofertilizer.
Good afternoon, everyone. I am Kanika Sharma. I have recently joined in PhD under the supervision of Rajgopal sir in HCL.
Good afternoon, everyone. I am working as a senior technical officer in the Ministry of Water Technology. My area of work is isolation of lactobacteria from diverse environments, and we are isolation of secondary metabolites from lactobacteria and biological evolution. And apart from that, I would like to pursue my research work in the algal research. That's why I used to come this.
Right.
Good afternoon all. My name is Monica. I am pursuing second year PhD under the supervision of Dr. S. Venkatamohan sir. My area of work is especially in electroculture, focusing on root rhizosphere with the influence of electricity in situ potential in molecular biology.
Myself Rajendra. I am currently pursuing first year of my PhD under the guidance of Professor Rajgopal sir. Currently, I am working with the desert algae Cestria and studying some of the biophysical properties of that algae. Thank you.
Hello everyone, myself Namrata Dubey. I am third year PhD scholar and currently working on abiotic stress, soil and temperature stress and investigating the role in javelin. Thank you.
Good afternoon all. I am Pavitra. I am working with Rajagopal sir. I am in my final year of PhD and I work with the modeler organism Vinoriella salina. It's a photosystem remodeling in hydrocolor.
I am Shreya Sharma. I am a third year PhD scholar under Professor Rajagopal and my work entails studying the redox control in Chlamydomonas radiata.
Good afternoon, myself Dr. Lakshmipati. I am doing as a postdoc researcher with Rajgopal sir lab.
So
Good afternoon, everyone. Myself, Dr. Venkateshwara. I work as PhD Inspector Faculty in the Department of Plant Science in the University of Hyderabad with Professor Rajagopal.
Good afternoon, all. Myself, Jerome, and I am in my fifth year of PhD under Rajagopal sir itself. So I work on chlamydomonas, particularly characterizing few autophagy gene, and with the interest to enhancement of lipid tolerance.
Good afternoon, everyone. I am Jyoti Ranjan. I am working under Professor S. Rajagopal. Means like my work is mainly focused on Poisson cell abiotic stress, mostly work on temperature stress and as well as fluctuating head stress.
Myself, Mohammed Sohail. I am working on Advanced lab. I am just completed master's degree. Then I just work as a project assistant in our lab, mostly working on the photosynthesis and Poisson sativa.
Kozami-san, would like to introduce yourself? Yes.
My name is Kazumi Takahashi. I am Yuichiro's wife. I was a school teacher, elementary and junior high school. But now I retire and I have a time to come India.
You are welcome. Okay, so Echiro, you want to say something or no?
I think I said already, but
Yeah.
yeah, nothing to add.
Okay, so said this the workshop is about the both lectures and the tutorials. So every afternoon you have a tutorial, right? You can see the program. All of you received program, right? You would like to have physical copy? It's fine, huh? You like to have physical 1 or 2 copy? You can make and give it to them. Whoever wants, you can collect from Jyoti or yeah, Jyoti will give you. So we have 24 talks, total 24 lectures and 7 tutorials. I guess 7 tutorials. No, 9 tutorials. Total 9 tutorials are there. Then except last day and all other days we have a tutorials. So each these 24 lectures. Now today we have 2 lectures, and tomorrow sometimes 3 lectures. And also Professor Raghavendra has taught, and as I said Professor Echiro Takahashi is a foreign faculty. I am also teaching some of the topics you can see, and here also we have invited Dr. Madan is going to teach, going to talk one of the topic accumulation process. And there is one Professor A.B. Das from Utkal University who also work on photosynthesis. He is here and he will be teaching 2 lectures, that will be day after tomorrow and afterwards. Day after tomorrow means tomorrow is 8th, 9th and 10th is going to teach 2 talks. And one more Professor B.C. Tripathi, probably in the program it is already there. So he is also expert in photosynthesis and chloroplast biogenesis in higher plant site. So he worked almost same thing like about 40 years in the photosynthesis related area. So he is going to focus more on chloroplast biogenesis in plants and as well as the gene expression related to chloroplast. So 13th and 14th, 15th he will be with us. You can also interact with him. And one more, the other talks my own student Dr. Yusuf Muhammad, who is going to take care of one, that is bacterial light arresting complexes. I also asked Jerome to take care of one of the talk, there is chloroplast transformation, one talk will be there, the rest I will be covering. So, the main, as I said, the main motto of this workshop is to train all of you in photosynthesis related topics, training in hands-on training. So, my students anyway, they do not need to train, but they will help you in training of all these whatever we have designed, and they will also be going to circulate all the the forms and on the not forms, the protocols. That protocols you can implement, you can go back and implement it in your lab. It works. So, first we will start with the chlorophyll fluorescence, that is a simple instrument, handy P, how to measure the photosynthesis. That is a chlorophyll fluorescence. Based on the chlorophyll A fluorescence, you get the fluorescence, that is photosynthetic activity can be measured. Then we also have a is called photosynthetic, the PAM mission, that is little more higher end, where you can differentiate photosystem 1 and photosystem 2 and even cyclic electron transport. All these linear electron transport can be measured, linear and cyclic electron transport, that can be measured, that will be one day. And most of them, those in India and wherever, when they want to study some tree physiology, I mean tree related photosynthesis or higher plant related, they use IRGA mission. So, IRGA will be also, you will be trained how to measure the photosynthesis, CO2, transpiration rate and CO2 uptake, and water use efficiency and photosynthesis efficiency, and this also can be, you will be trained very well. Further, since it is the chloroplast biogenesis, and we will be going little deep into more of biophysical and biochemical experiments. You separate all the complexes like thylakoids, the first one, chloroplast and thylakoids, then see what are all the complexes present in chloroplast. This very good one is called super complex separation by blue native gel. So, that our students Pavitra and I think the names are there. I mean the new program, can you share with them? So, all the names are there, who are going to take care of the experiments. You can interact with them even if you go back and you want to get more information or some kind of help, they will be helping you. And now the blue native gel, and we will also see some biophysical aspects, how the protein and pigment they interact. So, that using circular dichroism, only we will look at the UV visible side, visible range from 400 to 800 nanometers, that they will be also helping you to get trained. So, you can train, actually now that is the good that there is only 15-16 students here. So, you can have all of you can train very well if you are focused, I do not know if you are interested. So, and now if time permits, we can add many more, but we will see. Then we will jump on to the one topic is metabolome, analysis of metabolome, that is using we have already metabolites already, we have you measured, but you cannot measure now, it takes time. From that, how we can analyze metabolites, either chloroplast metabolite, any metabolites, that will be Dr. Yusuf will take care of one of the chapter, one of the tutorial. Then true molecular biology, chloroplast to cloning, right? Cloning, cloning and transformation and isolation also will be studying. RNA isolation is also will be 1-2 days, that will be also covered about the chloroplast and little more molecular biology. So, these are the the experimental setup. So, I request all of you concentrate on all those aspects. so that you will be really trained and it will be advantage for those who want to stay in this field. Of course, these techniques can be used anywhere, though unless, as long as if you are in a plant science, these techniques can be, you can apply, to check the physiology of the cell or physiology of the any leaf. This will be, really definitely it will be useful in future. So, the sometimes these whatever we said the strictly time, some time is meant, but even if it is late, you can stay a little longer and get you himself, you have to show your interest. None of my students will be more in, they will be active, if you don't show interest, they will also, okay, what is there, they are not showing an interest. So you have to show interest and they will be definitely work with you, or they will definitely train you much better. So we will also take help from Professor Echirosan if anything related, all these experiments. So that will be also, because you also can contact him anytime. You can get some information, any information related to whatever the topics which is going to talk, or whatever the topic, the tutorials which we are going to cover. You can also contact me or contact Professor Echiro. So these are some of the the kind of orientation I wanted to tell you before we go for tutorial. So we'll be using leaves as well as we'll also using cells, the chlamydomonas cells, to show you the function. Jyoti, going to use leaves also, right? Okay, leaves as well as the chlamydomonas, so that you can understand much better differential photosynthesis, if there is any difference in photosynthesis plant versus an algal system. So once again, all the best for all of you, and thank you for joining for the workshop, and you will definitely will have good time here. And few more things, few announcements. The GIAN conference is a very prestigious conference in India. Not everyone will get this award. It's like kind of award for me to the department. It's like once they in a play, it is like we they scrutinize and review, and they select the project. And they have mandates that you all have to give attendance every day. I think Raghendra is collecting attendance, and there is signature, right? Yeah, you need to sign signature. That's very important. I don't know what they do if you don't come. I have no idea, but as a coordinator, I have to submit those the records, and also your feedback about the conference or the workshop. You have to give fair, your judgment. There will be a grading. You can give the grading. Those gradings are also important. Improve from their side. Okay, if there is anything improvement, not paying money for these things about subject. Subject you need to be improved, or it could be much better, or some kind of anything improvement, so that they will improve, and then they will change the program slightly. So this is feedback, and attendance is mandatory by you guys. Okay, so all the best, Jyoti. Now you can start with the chlorophyll fluorescence, and you can measure yourself also. He will give enough time to you to measure, and also explain little bit the curves, OJIP curve.
Will plot it in computer.
Yeah, you can do it there itself. Connect this computer. Huh?
Yeah, I can do that.
Yeah, here itself you can do it. Where is Shailesh? Shailesh is also helping, no? Oh, she went there to bring the part. Okay, then go and bring the parts.
Suhail already went.
Oh, there, it's there. Okay, then. So all the best, and I'll be in my office. If you, I'll in between and come. I will also see the results. If any problem is there, just give me